Cytoplasmic matrix. Cell, whether prokaryotic or eukaryotic, contains a fluid, and this fluid is nothing but the cytoplasm. Various metabolic activities that take place by using this platform, that is cytoplasm. So here we will focus the things related to the cytoplasm. Actually, discovery of cytoplasm is not a credit of particular scientist, but is a collective, due to collective efforts of many scientists. What we know about cytoplasm is there. Cytoplasm is part of cell outside the largest organelle, nucleus. So surrounding to nucleus, in the liquid form, which substance is there, which is thick, gel-like, semi-transparent, and is found both in plant and animal cells, is cytoplasm. This cytoplasm is bounded by plasma membrane and contains many organelles in the cell. The constituent parts of cytoplasm are cytosol, organelles, and cytoplasmic inclusions. Cytosol, the aqueous part of the cytoplasm outside all of the organelles, also contains its own distinctive proteins. So how this cytoplasmic matrix in its nature, what is its physical nature? So we will try to focus on this thing. Cytosol or cytoplasmic matrix is colorless or grayish, translucent, viscous, gelatinous or gel-like colloidal substance. It is heavier than water and has capacity of flowing. So what is the physical nature of the matrix? To discuss this thing, different scientists, they have their opinions and they have put forth it in the form of theories. So one by one, what is the physical nature of cytoplasmic matrix, that we will discuss with the help of few theories. First theory is the granular theory. See here, cell cytoplasm is shown in the form of granules in it. So granular theory put forth by Altman in 1893, who is of the opinion that protoplasm consists of numerous tiny granules. For example, amoeba. Second theory is alveolar theory. Alveolar nature of protoplasm. It means just like having air sacs. So these things just here shown. Alveolar nature of protoplasm, it was suggested by Buckley in year 1892. So according to him, protoplasm consists of suspended droplets of or alveoli or minute bubbles resembling the forms of emulsion. Third theory, fibrillar theory, put forth by Fleming. So it proposes that protoplasm consists of fibers embedded in the inner mass of matrix. Embedded in the inner mass of matrix here. The fibrae are also called mytum or spongioplasm, formed of proteins called nacelles, and ground substance is termed paramytum or hyaloplasm. Fourth theory, to explain physical nature of cytoplasmic matrix, is reticular theory. See here. This theory proposed by Klein and Comay. It suggests that protoplasm consists of a reticulum of fibers in its hyaloplasm. Hyaloplasm means clear fluid portion of the cytoplasm. Organic material which consists of protein and carbohydrates in suspension may be either water loving, that is hydrophilic, or water hating, that is hydrophobic. The hydrophilic particles occur surrounding to the water molecules. The attraction between protein and water is due to electric charges that hold them together. Then fifth theory is latest one, and name of that theory is a colloidal theory. A recently forwarded theory, that is colloidal theory. So after electron microscopical observations of the matrix, this theory has been forwarded by the researchers. According to this theory, matrix is partly a true solution and partly colloidal system. Matrix is a partly true solution and partly colloidal system. Now solution is mixture of liquid called solvent and any chemical substance in solid or liquid called solute. So solution is the combination of solvent and solute. In solution, particles of solute should be less than 1/10,000 millimeter in diameter. Solution part of matrix consists of water as solvent, in which various solutes of biological importance such as glucose, amino acid, electrolytes, minerals, vitamins, also hormones or fatty acids and enzymes remain dissolved. And colloidal system, one can define colloidal system as system which contains a liquid medium in which the particles ranging from about 1 upon 10 lakh to 1 upon 10,000 millimeter in diameter remain dispersed. In solution, this particle size 1 upon 10,000 millimeter in diameter. In colloidal system, these particles size ranges from 1 upon 10 lakh to 1 upon 10,000 millimeter in diameter. Such particles, they remain dispersed in colloidal system. Thus, colloidal state is a condition in which one substance like protein is dispersed in another substance to form many small phases suspended in one continuous phase. Colloidal system consists of two phases: discontinuous phase, also called as a dispersed phase, and dispersion phase. Whole of the protoplasm, cytoplasm and nucleoplasm is colloidal solution because main molecular component of protoplasm, proteins, show all characteristics of colloidal state. Proteins form stable colloids because firstly they are charged ions in solution that repel each other, and secondly each protein molecule attracts water molecule around it in definite layers. This is what related to the colloidal theory. Now phase reversal. Cytosol or cytoplasmic matrix, like many colloidal systems, shows properties of phase. For example, gelatin particles, take it as discontinuous phase. These gelatin particles are dispersed through water, which is a continuous phase, in a thin consistency that is freely shakeable. Such condition is called sol. The solution cools, gelatin now becomes continuous phase, and water is in discontinuous phase here. Now solution has stiffened and becomes semi-solid and is called gel. In gel state, molecules of colloidal substance remain together by various types of chemical bonds. Maybe bond between hydrogen-hydrogen, between carbon-hydrogen, or between carbon and nitrogen. Stability of gel depends on nature and strength of chemical bonds. Now if one heat the gel solution, so due to heating, gel solution, it becomes sol again and phases are reversed. Under the natural conditions, phase reversal of cytosol or cytoplasmic matrix depends on various physiological, mechanical and biochemical activities of the cell. Chemical organization of cytoplasmic matrix or cytosol. What is chemical makeup of cytosol? We will study it here. Actually, cytoplasmic matrix composed of many chemical elements in the form of atoms, ions, and molecules. Chemical elements present in the matrix essential one are 24 elements. Naturally occurring 92 elements. Out of those, about 46 are found in cytosol. 24 of these considered as essential for life. We call them as essential elements. While others are present in cytosol only because they exist in the environment with which the organism interacts. Out of these 24 essential elements, 6 elements especially are very important and plays important role in the living system. These major elements are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Most organic molecules are built with these 6 elements. Another 5 essential elements found in less abundance in living system are calcium, potassium, sodium, chlorine, and magnesium. Several other elements, which we will call trace elements, are also found in minute amounts in animals and plants. These are iron, iodine, molybdenum, manganese, selenium, cobalt, copper, chromium, tin, vanadium, silicon, boron, fluorine, nickel, etc. After chemical elements, cytoplasmic matrix or cytosol contains ions in it. Cytoplasmic matrix consists of various kinds of ions. These ions are important in maintaining osmotic pressure, also acid-base balance in the cell. Retention of ions in the matrix produces an increase in osmotic pressure and thus entrance of water in the cell. Concentration of various ions in the intracellular fluid differs from that in the interstitial fluid. For example, in the cell, potassium and magnesium ions can be high, and sodium and chloride ions high outside the cell. Silicon ions occur in the epithelial cells of grasses. Free ions of phosphate occur in the matrix and acts as buffering system and tend to stabilize pH of cellular fluids. Other ions like sulfate, carbonate, bicarbonate, magnesium, and amino acids are also present in the cytosol. So after chemical elements, then ions, cytosol also contains electrolytes which play important role in maintaining osmotic pressure and acid-base equilibrium in the cytosol. Magnesium ions, phosphate are good examples of the electrolytes. Non-electrolytes are also there in the matrix. Non-electrolytes of the matrix are sodium, potassium, calcium, magnesium, copper, iron, manganese, zinc, cobalt, nickel, chloride, molybdenum, etc. Iron occur in the hemoglobin, also in the cytochromes, also in some enzymes as catalase and cytochrome oxidase. Calcium occurs in blood, matrix, and also in bones. Copper, manganese, molybdenum, zinc are useful as cofactors for enzymatic actions. Iodine, fluorine is essential for thyroid and the enamel metabolism.